Immunis has is it a product or a technology called Immuna, correct? It's
It's a
a tech,
product. Yeah, it's
it's
actually it's a new drug in a
right,
brand new drug category. It's called Immuna.
Immuna, right. Okay, so could we talk about so you said you're using human stem cells, young human stem cells. So can you talk about
Yep,
where are you getting them from and how are you
sure.
creating so much um, secretome from them?
Yeah. So we use a ethically procured, laboratory generated, NIH um, uh, registered. These cells have provenance. The starting materials are either iPS inducible pluripotent stem cells that are human or embryonic stem cells that are human, obtained from the discard of fertility clinics. The the world is you know split in some jurisdictions really like iPS cells. Some jurisdictions really like embryonic stem cells. So we can make the product out of both of them.
I mean, how often do you need to renew like the stem cells?
You really don't. It's
Yeah.
they like a an embryonic stem cell divides very, very well. It's naturally occurring, it's very young, and once those things are isolated, they can last hundreds and hundreds of years of dividing. You could, in a single stem cell line, if you didn't stop it from growing, the earth would be covered in about a 12-foot slurry since they were first developed in the late 90s, or isolated, I mean. But you know,
Mm.
uh, uh, iPS cells, when you take human skin and then bombard them with a whole bunch of factors that
Mm.
genetics that are expressed by young cells, kind of tricks the skin cell into thinking that it's a younger embryonic cell, and it back differentiates and becomes a young stem cell. So it's another way of making it. But the
Mhm.
means of making it, jamming in 1,500 copies of four different things, beats up that cell. So the cell is a little less capable of dividing. And so it'll stop. It might do 10 or 20 divisions and then start to differentiate and you lose your pool. Whereas embryonic stem cells have a much more robust ability to stay young. And therefore the banking of them is really one cell has uh, really, you know, formed the foundation of almost every treatment that my laboratories have ever generated in the last 30 years.
And actually, you're not using the cells. So you keep the cells. The cells are like a factory that are generating the secretome and you're extracting the secretome. Would that be correct?
Yeah, that's exactly right. It's very interesting. We are, in a brand new uh, drug in a brand new drug category. It's very fun at Immunis. The 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 cells that we start with are stem cells, and then we make these young precursors that secrete this immunological modulator soup, if you will, of 440 factors, the secretome. And we just keep farming the cells, but the product is actually just the proteins, the secretome, what is secreted. So there's no self or non-self on those things. They don't get rejected. So therefore, this is off the shelf. It is something that you can just use from anybody because your immune system factors are like mine. My TGF beta is like yours, my IL-10, my IL-2. They're so similar across genders and races, ethnicities, that we can actually make them. It's called recombinant proteins synthetically and use them across genders and ethnicities. So the getting them from one particular cell line is good. Basically, they're not rejected by any other individuals. So it's off the shelf. It's a secretome that has 440 active ingredients that targets the immune system in 440 different ways. And that is really the special bit of what we're doing. The immune system is so complex. It is so incredibly complex. I'm a neuroscientist. I thought the brain was complex. Started beginning into immunology about 15 or 20 years ago. And my goodness, it is so, so complex. For every physiological thing that your immune system does, there's 5 or 50 things pulling and 5 or 50 things pulling. It's like an orchestra that has a beautiful, harmonious output, but it's a lot of work to get that output. And drugging that with a single active ingredient like like every drug you've ever taken in your life. Every drug is a single active ingredient. But back in the days when botanicals were coming up and lots of good medicines were being made from plants, the problem that the FDA had at that time is batch to batch variability. You know,
Mm.
you boil a pound of willow bark and you get a different amount of aspirin derivative every time. So it's hard to make. It's sort of like a terroir in wine. So as you know, botanicals are not generally regulated by the FDA because of the batch to batch variability. Whereas cells, the the developments in cell culture have been happening in leaps and bounds over the last 20 years. My laboratory at the forefront of this, and we're perhaps the world's best at growing homogeneous populations of stem cell derivates, meaning we take stem cells, make them into something in purity. Oligodendrocytes, a spinal cord cell type that's missing after spinal cord injury. You make that in purity, and it restored, you know, quite a lot of function to quadriplegics in clinical trials. We've made high purity cardiomyocytes and neurons and neuronal progenitors. And in this case, a cell type that's young and secretes every factor relevant to immune system development and health. It's an interesting new drug category, the secretomes, just a few years old. I believe that Immunis is only the second company in the world that's making them, or in the United States. Excuse me. And 440 actives in one drug.